Lab'da testlere bakıp çantamı alıp geleyim. Tamam, biraz bir şeyler.
Serap Hocam merhabalar, hoş geldin.
Hocalarım nasılsınız?
Teşekkürler, sen nasılsın hocam?
Teşekkürler, sağ olun hocam. Öğrencileri de organize ettik. İnşallah katılım yüksek olacak. Umut ediyoruz.
Çok teşekkürler. Evet,
Rica
çünkü niş bir konu, tam da sizlerin ilgisini çekecek bir konu hocam.
Evet. Lisans öğrencilerimiz
Serap Hocam buradayız.
dinliyor. O gelmişler. Güzel.
Kameralarınızı da açarsanız çok seviniriz. Sevgili Serap Hocamızın ekibi.
Hemen hocam saçımı, üstümü, başımı ayarlayıp hemen açıyorum kameramı.
Misafirimiz de burada, Gonca Karahan.
Merhabalar Gonca Hanım.
Merhabalar. Benim sesim bilmiyorum ama.
Önceki haftalardan farklı olarak bu haftaki misafirimizle aynı toplantı odasından katılıyoruz. Birimiz mute edince öbürümüz konuşacak şeklinde. Bakalım o ayarlamayı umarım yaparız. Kutsal Hocam merhaba, hoş geldiniz.
Zeynep Hocam merhabalar, hoş bulduk. Nasılsınız? Herkese
İyi,
tekrar
sağ
hoş
olun.
geldiniz
Siz de
diyorum.
bir saçınızı başınızı düzeltince hocam.
Hocam ben hazırım. Açayım hatta.
Evet, herkesin kamerasını açmasını bekliyoruz. Kamerasını açmayanı kabul etmiyoruz şimdi, evet. Öyle mi? Bizim hocam da geldi.
Herkese sağlık.
Türkçe'ye mi dönüşünüz var Gonca Hanım?
Dönesim yok yani zaten. Ben istediğiniz değil, istersen bir değil.
Şöyle. Evet, isterseniz yavaş yavaş başlayabiliriz. Evet, herkese iyi akşamlar. Hoş geldiniz. Oradan mı giriyorsunuz hocam? Tamam, tamam.
Evet değerli arkadaşlar.
Ben kapatıyorum o zaman.
Tamam, sesim geliyor mu? Ben de buradayım. Evet değerli arkadaşlar, akademisyen arkadaşlarım, öğrenci arkadaşlarım, yüksek lisans doktora yapan arkadaşlar, Fen Bilimleri Enstitüsü'nün yaptığı bir konuşma serisinde tekrar beraberiz. Ben hepsini takip etmeye çalışıyorum. Gerçekten çok değerli konuşmacılar sizlerle birlikte oluyor. Bugün de aramızda çok değerli bir konuşmacı var. Gonca, hoş geldin. Yanında Gonca. Hollanda'dan, Leiden Üniversitesi'nden geldi. Çok özel bir konuyu, tecrübesini bizlerle paylaşacak. Kendisini ben eskiden beri takip ediyorum. Hollanda'da da ziyaret etmiştim. Orada da ağırladı bizi. Bilimsel derinliği çok yüksek. Türkiye'nin yetiştirdiği değerlerden ben hep onu söylüyorum. Yıldız Teknik Üniversitesi'ni transfer etmek için de çok uğraştım. Ama inşallah onu ikna edeceğiz. Türkiye'ye tekrar geri dönmesini sağlayacağız. Keyifli bir konuşma olmasını diliyorum. Gonca'ya teşekkür ediyorum. Zeynep Hocama teşekkür ediyorum. Murat Hocama teşekkür ediyorum. Emeği geçenlere teşekkür ediyorum. Fen Bilimleri Enstitüsü'ne teşekkür ediyorum. Bunların da çok önemli bilimsel toplantılar ve buluşmalar olduğunu düşünüyorum. Hep bir sloganımız var. Sadece derslere girmekle üniversitede olunmaz. Sadece derslere girmekle yüksek lisans doktora yapamazsınız. Ders dışı etkinlikler de çok çok önemli. Test çalışmaları çok önemli. Bu buluşmalarda bunları besleyen şeyler. Tekrar hayırlı olmasını, verimli olmasını diliyorum. Sizleri sevgi ve saygıyla selamlıyorum. Çok sağ olun. Muratçım buyur.
Hocam çok teşekkür ediyoruz değerli katılımınız için. Sizi de burada misafir etmek bizim için olur. I'm going to switch to English actually from now on. Today we're going to have a guest, Gonca Karahan. She is a researcher from Leiden University, and actually she is a very old friend of mine. She's a very old and close friend of mine. Today's seminar topic is going to be transplantation immunology. Actually, we're going to talk about how we're going to find the perfect match. She's going to explain us how we're going to find our perfect match. Okay, first of all, actually, I want to have a brief introduction of our invaluable guest. Gonca Emel Karahan, Dr. Gonca Emel Karahan. She is a postdoctoral researcher in Department of Immunohematology and Blood Transfusion in Leiden University Medical Center. She was, I don't want to say when she was born actually, but after graduating from Beşiktaş Atatürk Anadolu High School in 1996, she started her studies of biomedical sciences in Cerrahpaşa Medical Faculty in Istanbul University. From her graduation in June 2000 until December 2011, she worked at the Diagnostic HLA Laboratory of the Department of Medical Biology, Istanbul Medical Faculty, which we know it as SAPAGA, right? During this period, she received her master's degree in 2003 and PhD degree in medical biology in 2010, with research focused on the detection of tissue antigen-specific antibodies in kidney transplant recipients. This part is interesting. After completing her PhD, she didn't give up. She decided to make another PhD. I don't know why, but she did it. In January 2012, she moved to the Netherlands for her second PhD education in the renowned Transplantation Immunology Laboratory in Leiden University Medical Center, where she carried out research on tissue antigen-specific memory B cells. Since her second PhD graduation in 2017, she is working as a postdoctoral researcher in the same group, extending her research to HLA antibody characteristics and overseeing the research and development of the HLA diagnostic laboratory. It is a very great pleasure to have you here, Gonca, and I am giving the word to you. Thank you very much.
No sound. Sorry.
Sesim geliyor mu diye bu konuşmaya başlayayım o zaman.
Yes, okay.
Okay. So I want to say also a couple of sentences in Turkish and then going to switch to the format in English. Herkese merhaba. Öncelikle böyle bir daveti aldığım için yakın arkadaşımdan çok mutluyum, çok seviniyorum. Tamer hocamıza da aynı zamanda bu fırsatı verdiği için teşekkür ediyorum. Zeynep'in de bahsettiği üzere ben organ nakli immünolojisi alanında çalışan, onunla ilgili araştırmalar yapan bir insanım. Buradaki katılımcılara, genç arkadaşlarımıza eğer araştırma alanında kendilerini geliştirmek için yurt dışına giderlerse belki bir nebze olsun ilham kaynağı olmak adına sunumumu düzenledim. Belki biraz fazla sıkıcı noktaları olacaktır ama umarım yılın bu son gününde daha böyle hafif bir konuşma şekline dönüştürebilirim. Now I'm switching to English and going to share my screen and ask you also, do you see my screen? Do you see it? It's great.
Yes.
It's always wonderful to be heard and to be seen in Zoom meetings. So if I can also find my, okay. So as Zeynep mentioned, I am now working in Leiden University Medical Center, and I focus my talk on transplantation immunology. So I'll try to actually give a brief understanding of what actually transplant immunologists do in order to find the best organ donor for a patient. So I gave my title Finding the Perfect Match, which I think also gives you an understanding that it sounds like finding your soulmate, finding your love in life. So I'm going to start actually my talk with an experiment called the sweaty t-shirt experiment, which was also a subject of a BBC documentary in 1990s, where they investigated whether the mate choice, the partner choice in women was dependent on some biological factors. So in this experiment, they asked university students, women, to smell t-shirts from 7 different men. And they asked them which of the smell from which of the t-shirts they found attractive. So in this, let's say, experiment, women actually chose the smell of the men in the t-shirts, those that had the most different immune system when compared to their own immune system. So the smell plays an important role in selection of the mate for the woman. And the ones that women found, let's say, fine as not bad smell of the t-shirts was those that had the most different immune system than theirs. So this is, of course, it sounds a little bit like a magazine news, but the hypothesis under is that women want to reproduce with the most surviving genes. So in order to have a child, in order to reproduce in a healthy way, they want to have the most different immune system so that the child can have some of it from the father, some of it from the mother, and combat with variety of viruses. So I find this very, very interesting because in this experiment, let's say, the women preferred to mate with the most different genetic makeup owning men. So this may sound a little bit maybe too magazine, but this is actually a scientific paper published in 1995 by a Swiss researcher in a scientific journal. And as you can see, let's put a pointer also. As you can see here in this graph, here they asked these women to score the pleasantness of the male odor in these t-shirts. So they are candidate partners in life, so-called, and they said that they found the smell of the men who had the most dissimilar MHC type. So this MHC is now going to be the focus of my talk. This is actually a molecule, a set of molecules, a set of large set of genes that are coded in the major histocompatibility complex. If you look at what this histocompatibility means, it is actually the matching of the tissue. So these are the molecules that we have in our immune system that are essential for an adaptive immune response. So I understand that some of these terms might be very, very complicated, but when I say adaptive immune system, it means this is not the immune system that we are born with. This is not the innate one. This is the one that we build throughout our lives by combating infections, combating pathogens, and then we build on top of it. We create an immune system which we call adaptive immune system. And these molecules, which are called major histocompatibility complex proteins, in humans we call them human leukocyte antigens. So they are present on our white cells. So they are called tissue antigens, and they are variable among all of us. So I always compare these molecules to blood group antigens. So we have A blood group, B blood group, let's say O and AB type, simply 4 blood groups. Of course, there are some subtypes, but we have there are many human leukocyte antigens, tissue antigens, and they identify who we are, who our immune system is. So these molecules are very necessary for us to recognize the pathogens, and they are very, let's say, disparate between individuals, but actually the molecules that we use, let's say, to find our partners in life are a problem, a barrier in transplantation when we need an organ from these people. So as said, my talk will focus on now how these important molecules which we use to defend ourselves against microbes or pathogens are a problem in transplantation. So you may all be aware that nowadays it is possible to have kidney transplantations, liver transplantations, heart transplantations, so solid organ transplantations, but also hand and face transplantations, uterus transplantation also possible in our country here in Turkey. So there are several different types of transplantations possible, in addition to also bone marrow or stem cell transplantations. My focus in my research is mainly on kidney transplantation, which is the best treatment option actually for patients who have an end-stage kidney disease. So this is also a graph, maybe it's from America, United States, but this picture is same in every country, in my experience, at least in Holland and in Turkey. Here we see in black arrows, the number of patients waiting. So there is a huge number of people in the world waiting for an organ, let's say a kidney in this talk, kidney transplantation. However, neither the number of transplants nor the number of donors are enough for those patients. So there is a shortage of organ. Therefore, it is very important that we make a nice risk estimation before we allocate organs, because in disease donor transplantation, if one person donates organs, 8 other people can have their life saved with the organs that's donated from one person. So we have to be very careful, people working in this field, who is going to receive this organ. So finding the recipient, finding the patient who will benefit more from the donor organ is called risk assessment. So these molecules that I have mentioned, human leukocyte antigens, HLA, are the core of this immunological risk assessment. This is the field that I've been working on for almost 20 years. So let's come back to this woman who smelled the t-shirt and found one of the men to be her husband. They decided to build a family. They made 2 children together, and now they are a family with 2 children. This woman, years later of her last pregnancy, can become an end-stage renal disease, end-stage kidney disease patient. And one of the treatments, one of the therapies for these people who do not have a kidney function is hemodialysis. So these people go every week, 3-4 times to dialysis centers and stay there 5-6 hours, and they replace their kidney functions by being attached to these machines. Of course, this is not the optimum way of life. This is not the standard of life that a person should have. So actually, kidney transplantation is the best treatment option. Such also other types of transplantations is the best type of treatment for patients with organ deficiency. So of course, the loved ones would like to give their organs in most cases. And in this case, the husband says, hey, I'm going to give one of my kidney because, as we know, in healthy individuals, most of the time we have 2 kidneys. We can donate one of them. So the husband says, okay, I want to give my kidney. Now, my question is, this man is a perfect match for this woman to have a family, but is his kidney also the perfect match for an organ transplantation? Now, I'm going to show you what we do as transplant immunologists in the labs to see who is the best donor for a patient. And I'm going to touch on the research that I've been working on in order to contribute to this risk assessment. So in this risk assessment, immunological risk assessment, HLA laboratory plays a very important role. This is a diagnostic laboratory that serves for the benefit of patients who are preparing for either solid organ transplantation or bone marrow or hematopoietic stem cell transplantation. But for kidney transplantation, it does several tests. It looks at the matching, the compatibility between a patient and a potential donor. It also screens the patient if there is something that's going to already harm before we put the kidney in the patient's cells, let's say. So these laboratories are very well, let's say, controlled and assessed by European or American federations in all around the world. And actually, I have started my career after graduating from biomedical sciences in one of these first European Federation of Immunogenetics accredited lab in Istanbul Medical Faculty, known as Çapa Tıp Fakültesi in our country. So I worked there from 2000 to 2011. This is the first lab in Turkey that had this accreditation, meaning giving service to patients at a high quality level. So wherever you go in the world, if a laboratory has this accreditation, you will get the same quality service everywhere. So it's standardized. So this laboratory is also not only the national reference center for such matching between patients. It's also the first bone marrow bank that was generated in Turkey. So I worked there 11 years. I did a PhD there, but I want to specify now why I did a second PhD. So in this department, I was hired as a master's student and a PhD student, but our first aim was to be involved in the patient-oriented work. So the HLA laboratory work, assessment of the risk for patients was our primary goal during the day. And after that time, we were focusing our research. So this gave me enormous knowledge on how this risk assessment is done in real life, daily life. It also increased my connections with the clinicians, which is very important. But my interest was more into research. It gradually came to a point that I would like to do something more research, but still patient-oriented. Therefore, after finishing and working for 11 years in this department, which I still have close connections, of course, I moved to the Netherlands, where there is a research lab and also an HLA laboratory. So there, the responsibilities were more towards the research lab and less towards the HLA laboratory during my PhD. And this was also an accredited lab, as you can see now. So the research lab in the Netherlands that I'm working, that I've started my work, is called the Transplantation Immunology Research. It is a research group that's embedded in Department of Immunology, where immunology of tumor, immunology of autoimmune diseases are also investigated, but this is not my group. The group that I am a member of is the Transplantation Immunology Research, which is has a very close connection with reproductive immunology. What is reproductive immunology? Reproductive immunology is the research line where we try to understand how a healthy pregnancy, is not perceived as a non-self by the immune system. A baby is actually half strange to the mother, but in a healthy pregnancy, in most cases, the mother never rejects it as it would an organ from another person. So if we can understand, we say in our department, if we can understand how a pregnancy works, we can maybe also help transplant patients. We can maybe also understand how their immunological mechanisms work. So as said, this is also attached to a diagnostic laboratory lab. So in my opinion, for an efficient research, it has to be also in close connection with clinics. So as you may also understand from this, I think, scheme, my research is the research that I'm working on is not a fundamental research. So we are of course interested in how things happen, but we don't really try to understand if a molecule is involved in the mechanism of an biological event. We more try to develop methods or algorithms that can be used in patient-oriented settings. That's actually what my research focuses. So it's what we develop in the research lab. We should be able to use it for the benefit of patients in the diagnostic lab. So my position, since my second PhD in Holland, is focused in between the research lab and diagnostic lab. So I try to make a bridge between the research and this diagnostic lab for the benefit of the patients. So as I said, this is all focused around this transplantation antigens. In our research group, we try to see how can we best match a patient and the donor so that we prevent them to become high-risk patients. We also try to develop methods where we can understand how risky is this patient to be transplanted immunologically. We try to improve algorithms to allocate organs, and also we are working on treating high-risk patients. So can they be also lower risk and transplanted? But my focus, my research focus in this group is on developing immunological assays to better define the risk assessment. So if we can do a good assessment of the risk beforehand, then we can actually maximize the utilization of the organs, is, let's say, the motto. So then what is this HLA? I've mentioned it before. It is the ears and eyes of the immune system. The immune system needs to understand what is non-self and what is self. So it does this by the help of these molecules. They're called human leukocyte antigens. You can think of them as like waiters presenting a plate. In this plate, there is the piece of the pathogen. And these molecules says, okay, now this is the pathogen you need to get rid of. It tells the adaptive immune response the non-self molecule that the immune system needs to get rid of. In simplistic terms, of course, if there are friends here who are in this field, they will understand that there might be, of course, better explanations, but I'm trying to make it common for everyone. So this adaptive immune response is actually what we need for specific defense. So if we have a viral infection, if we have a bacterial infection, we like this response to give a response. We would like our body to understand that this is the target that we need to get rid of. So HLA molecules enables this recognition. It is very specific immune response, and it also remembers. So nowadays with the vaccination studies and stuff, the aim is all to have a memory response. So when I see this antigen, when I see this microbe again, my body should remember it and then give a better, stronger, and a more effective response. So that's why adaptive immune response that we develop in our lives is very, very important. So these molecules, the HLA, which enable this adaptive immune response to recognize the pathogen, to recognize the non-self that needs to be destroyed, is inherited from parents. So it is, we know that the blood groups are also inherited from parents, but HLA is codominantly inherited. So this is the mother cell, let's say. We have these HLA molecules with blue, and we have the yellow with the HLA molecules on the mother cells. And we have the father that has this. So, of course, this is a marriage that's not intrafamilial. So there is no relationship between the mother and the father. This is important because this may lead to another, let's say, phenotype. But here, mother and father, they have their own specific HLA. So these HLA molecules define who we are actually. So as I said, the child gets half of it from the mother and half of it from the father. So we as children, if our parents are not relatives, we will be always at least half, 50% matched with our parents. And the children, they will always get one from the mother, one from the father. So they will have like 25% chance to be matched with each other. So this is important. The more variability we have, the more a wide range of viruses, pathogens we can fight. So each HLA has a different, let's say, binding groove that can fight with a different virus. That's why women want to mate, women want to reproduce with men that have a different immune makeup, let's say, so that they can transfer transfer a diverse immune system to their children to combat better with the pathogens. So this is a very, very interesting area, because how are these molecules? So I showed you here, this is A1, B8. So there is a special coding of it, which I'm not going to get into details, but it's actually numbered according to the order of discovery. So they first found the HLA A1, so they called it A1. Then they found A2, they called it A2. And this happens around 1950s. And in the first international workshop, they identified 27 HLA molecules. So between individuals, there can be maximum 27 different HLA. But as of September 2020, we see that there are almost 30,000 different HLA alleles. This means it is very impossible, very difficult to find a complete match between two individuals. So not only the number of molecules, of course, increased these days, the technology has also increased, now improved. So now we can define up to 6-7 digits, something that was called A2 before 30 years ago. We now know all the details of this HLA molecule. So it is a very specific identity that's very specific to an individual. But this is a problem in transplantation. So it's good. We all have different diverse HLA systems, so we can fight against different viruses, variety of viruses. That's also why in marriages, in societies where intrafamilial marriages are more common, there are more diseases, by the way. But it's a problem in transplantation because, as you can see also here in the graph, if there is too many mismatches, too many disparities between a patient and the donor, the graft will survive less compared to those patient and donors that have less mismatches, let's say. Because all these mismatches is recognized by the body as non-self. And we know that the non-self is most of the time destroyed by a healthy immune system. So, but people also need organs from other people. We cannot always have a twin sister. We cannot always have a fully matched mother or father or sister, as I've mentioned. So patients are transplanted despite the mismatches between the donor. But of course, the technology, science has improved so much. The immunosuppressive drugs also, the drugs that are suppressing the immune reactions have also evolved. So when patients are transplanted, now they are also relying on a handful of immunosuppressive drugs. These drugs have, of course, side effects, but compared to the cost benefit, the life standard that's provided by a kidney transplantation for a patient, all patients are happy to take these immunosuppressive drugs to have a functioning organ. So it is, as said, despite these immunosuppressive drugs, so despite the fact that the immune system is blocked, suppressed, at least to have a rejection to not accept the organ, sometimes immune cells still recognize these mismatches as non-self and they want to defend against the organ. So we call these defense response in this presentation antibodies, okay? So when we transplant an organ, it works, but if an antibody develops or if the patient has already this defense before transplantation, this organ will be lost due to an immunological reason. I want to also specify, of course, there is a surgical aspect of transplantation. So these transplantations can be also have complications due to surgical reasons, or can have complications due to the underlying disease. But here we focus on the immunological reasons. So this defense mechanism may lead to the failure of this organ. So until now, I hope I'm making a little bit of sense. Am I making some sense?
Yes.
Yes?
Sure.
Okay, so I have a question here.
I am taking notes from the beginning.
Very good. Okay, so
these kinds of questions.
Very good. So
Gonca, ask us.
yes. No, I want to mention that we inherit our HLA molecules from our parents, so that's very, very important. Just underlying. I'm not going to ask a question yet. So, okay, so if the body recognizes the transplanted organ as non-self because it has some molecule HLA markers, let's say, that comes from the donor, it may reject it. In the earlier times when the laboratory methods were not that good, this was happening actually already when the patient was on the table. We call it hyperacute rejection. So because the technology was not good enough to detect these defense mechanism in the patient already beforehand, so patients were transplanted and then the antibodies were attacking the donor, and as you can see, it turns black immediately on the operation table. But nowadays there is technology to see it, and this almost never happens in ours, but it does happen still after some time. So this is just to give also some scientific touch that this is the proof of what I'm saying. So here we see the graft survival. So how long does it survive? Here we see that these patients are followed up 11 years. So considering that they did not have any defense mechanism beforehand, if they develop it after transplantation, as can be seen with the red ones, the ones that develop these antibodies, only 50% of them have a functioning graft. But if they don't develop any defense, then the organ stays with them, like in all of them, more than 10 years. So actually, the survival of the kidney transplantation 10 years is quite good if a nice immunological risk assessment is performed before the transplantation takes place. But it's not that only when you get a transplantation you have these defense mechanism. If you have a blood transfusion, for example, for another reason in your life, or if you get pregnant, you also unfortunately develop this defense mechanism against these tissue antigens. Yes, so these are sort of the unlucky part of the patients that have this history of a previous transplantation or a blood transfusion. And unfortunately, of course, because these patients have antibodies already before transplantation, if you give a donor organ, this antibody will bind to this tissue antigen on the donor organ, and the organ will be rejected. So it's very important that when we have a patient, let's say in the transplantation laboratory, we need to define if this patient matches well with the donor, and does this patient have antibodies against this potential donor organ that we're going to transplant. So I'm going to skip these parts, but this is actually a very, very big problem. So 20% of the patients in the Netherlands, but also in Turkey, around 25%, maybe even more, because we tend to have more children in our families. And yeah, almost all women who have children have antibodies against the half of their child that comes from the father of the child. So these people are a little bit less lucky in finding a compatible donor. So in Europe, there are prioritization programs to allocate these organs, first of all, to these people if it matches. And despite these special allocation, for example, as you can see in blue, after 2 years, only 60% of the patients can be transplanted. So 40% of them still stay on these waiting lists, and unfortunately, they have a very high mortality rate, meaning they die while waiting for an organ. So this is, of course, not only a very burdensome thing for patients and families emotionally, but it's also quite a burden on the health system. So renal transplantation is also more cost effective compared to these dialysis and renal replacement therapies. So as said, the HLA laboratory plays an important role. It looks if the patient and the donor is nicely matched, and it also looks at the patient's serum if there is any defense already generated because of this previous history, a transplantation or a pregnancy or a blood transfusion. Yeah. So when we expose ourselves by these means to a stranger HLA, the body probably will develop a defense mechanism. So this is a problem when it comes to transplantation. Normally, this is a good thing for combating, let's say, pathogens. So I gave here some examples in a simplified way how we do this in a transplantation immunology lab. So in a very simplistic view, this is the HLA typing of the patient, okay? So considering that these patients and donors are blood group matched, as you can see in its simple form, it's already very complicated. So there are all these numbers and we have 2 donor candidates for this person. Donor 1, matching, again, very simplistically is done like this. If there is a similar number, it's a match. If there is a different one, that's a mismatch, okay? So this donor 1, the candidate, has 3 mismatches compared to the patient, okay? We have another donor. Now this is, let's say, the other sister or the husband. And this husband or this donor is also 3 mismatched under these conditions, A1, B8, DR10. So which one are we going to choose is something that's important. Of course, there are many factors, the age, the physiological condition, the clinicians take care of it. But as the transplant immunologists, we look immunologically which one should be chosen for this patient. So, well, we first do if there is already a defense mechanism after this matching. So we take the serum of the patient. There is this technology now, which uses very small beads, synthetic beads coated with a panel of these HLA molecules. And if there is already a defense antibody against this HLA, we can see it like in this readout. I mean, it's not very complicated in my view. When we see this result, we say, okay, this patient has a defense against B57. Okay, this is positive, this is negative in a simplistic way again. So I combine now all this information. I have 2 donors that are equally matched for HLA. One of them seems to be not good because my patient has already a defense against it. So again, from a very simplistic view, but it's correct, then I don't take this donor. This donor can give to someone else that can benefit for it. For example, if there is a domino transplantation or a pool of donors, then I will take this donor because I did not find anything with this current diagnostic tools that reminds me to not choose this donor as a transplantation immunologist. So we give this advice to the clinicians and the clinician then takes the next step. I just want to underline that, of course, nowadays it is possible also to transplant patients despite the fact that they have antibody responses already. There are treatments called desensitization treatments. So doctors remove these antibodies and then enable a window of opportunity for transplantation. But this is, I think, not the top. This is not the topic of this current talk. So, but just want to mention that the technology has improved in a very positive way. So now I'm coming back to my first slide. Now, this woman found her love, decided to make a family, and unfortunately became a kidney patient. And now I'm asking, should the husband be the donor for this person? What do you think, Zeynep? Do you think we have enough tools to decide this? This woman is now a pregnancy. She has a history of pregnancy. So we know that half of the child comes from the father. So the woman might have already a defense against her half of her child. This defense is specially regulated in pregnancies, which we don't understand. But we know that it's not that good regulated in transplantation. So maybe if we give an organ to this woman from the husband, this woman will have a reaction against this. But can we see it with the current tools is where my research starts. So I want to now take a gear back and look at this graph. You don't have to answer. So this was a rhetoric question, yes. So if we might have seen these graphs also in nowadays because now it's very popular. But this is the antibody response. So if you look at this red curve, the first curve, this is the response we generate against an antigen, against a virus A, let's say. When we are exposed to this virus A, we generate this response, and then it fades away by time. But once we are infected again with this same virus, this time the immune response is very quickly getting its peak. And as you can see, the peak is more than the first peak. So this second response is very quick and more stronger than the first response. And we say this is also due to the memory response. So the body remembers, I have seen this virus, I have seen this microbe before. Now I have also already developed some immune response to this. I can reactivate my response to this and have a better, quicker and stronger response. So memory is very important because it quickly reactivates itself. So when it comes to transplantation, unfortunately, it's again a problem. Yeah, so we have these, when our body sees somebody else's tissue antigens HLA via blood transfusion or pregnancies or previous transplantations, we create this defense mechanism. And all the tools currently available in transplantation immunology labs are actually looking at these serum antibodies, okay? But when we see somebody else's HLA, when we have this pregnancy or when we have this blood transfusion, we may also create memory B cells. So these cells are silent cells. They don't immediately produce this defense mechanism, but when they see it the second time, they will very quickly become activated. And actually, we call it anamnestic response. So in the case of this pregnant woman, which we might not find any serum antibodies with the current tools, we may actually find some memory that can be reactivated. So my research, my efforts focused on developing an assay. There was no assay until recently that can detect actually these silent cells. So I'm not going to get into the details, but by using this assay, we think, and we have also done pilot study, we can have extra information than what we can have with the already available tools. So we don't have to understand all this graph, but if you focus on the red bars, the blue bars are what we know from the current diagnostics. So according to this woman, does not have any antibodies to DR10 or DR14, but if we use this memory screening assay, we can see that it actually has antibodies against DR10. So if you transplant based on the blue, you will see that there might be an anamnestic response deriving from the reactivation of these memory cells. So now the aim is, now this is what the HLA laboratory does some tests, immunological tests. Our aim is to add the HLA memory screening for the benefit of transplant patients, not to everyone, but those that are high risk, those that have this immunization history via blood transfusions, pregnancies, those women that are planning to have donor organs from their husbands, from the father of their children. And recently, Dutch Kidney Foundation has supported us with some funding to see whether this is really clinically relevant. So this will enable us to actually see if this memory assay we've developed is going to help us to better the immunological risk stratification in combination with the current tools. So this is, of course, a team effort, and I'm very, very grateful to everyone who helped and supervised me. And thank you for your attention. Mm-hmm.
Okay, Gonca, thank you very much for this really good presentation. I was actually thinking that it was going to be like Chinese to me.
Was it?
But it wasn't. But it wasn't. It was a really clear presentation for those actually doesn't know anything about transplantation immunology. So everything was quite clear. But it was quite difficult to respond as well. Okay, so we can skip to the questions and answers part if you like.
Sure.
You may raise your hands or you may write to the chat part of Zoom. If you like, you may ask your questions in Turkish as well. So please be comfortable to ask your questions. Okay. Okay, sure. Murat Hoca.
Okay. First, I would like to thank, I'm not from the field, but the presentation was very smooth and clean. So I'm very thankful. I understood almost everything smoothly. The thing is, I have a question from that tissue allocation, the donor and the patient's match. You said I don't take my note. HLA stands for, I don't know it, but you have sublevels A2, A11, and you are checking if there's mismatches or not. So imagine that there are two patients and we have three mismatches. Do we have the priority level for this A2 and A11? For instance, like you have to match the A2 prior to A11, or they are equally risky or equally likely?
Okay, can you hear me?
Yes.
Yes. So it's a wonderful question, by the way, and I'm not joking. So, of course, I could not get into the details of it, but normally some of these mismatches have some parts that are also similar to the self. So there are algorithms that defines in our also lab, and also it's being used, that defines like this is more preferred than that. So I cannot say, of course, right now from the top of my head, this is this, but there is an algorithm that says, okay, this is the mismatch load between this donor and this donor, so that we can also select. So what I show was very simplistic, but that's very relevant. In the older times, there were actually studies where they looked, hey, these are taboo mismatches, so never transplant from these mismatches. But nowadays there are algorithms that automatically calculate which is more preferable.
May I ask a question to you?
Thank you very much, Murat Hocam, for your question and the answer. Gonca, we can have the other questions. Is there anything written in the chat part? No. Kim? Göremiyorum. Nilbeste. Nilbeste. Yes, we're listening. Nilbeste, are you going to have a question? Yes, Mirai Bombom. May I ask one? Is the memory screening assay? Nilbeste, if you like, you can ask your question. You can speak.
Can you hear me?
Yeah.
So yeah, if you can hear me, thanks for your presentation. The memory screening assay you've mentioned before, your own procedure, is it based on the flow cytometry or is based on the ELISA?
So okay. So this assay that we have different formats. So one is dependent. One is an ELISpot based assay, so not ELISA but ELISpot, and there is also flow cytometric approach. But we have all these tools available. But this recent one that I have mentioned is Luminex based. So we stimulate the cells. We activate them in vitro polyclonally. We take the supernatants, we isolate the IgG, and then we use this antibody detection system, which is Luminex bead-based assay that we use for serum. We use it for these supernatants and see if there's a profile. But normally it can also be done in the older, let's say, days by ELISA, ELISpot, or also flow cytometry. But this depends on the question we are asking.
Okay, thank you so much. And one more question. Because of my husband's job, I'm spending a lot of time in Leiden, actually. So we have a home there. Can I spend a visit to you? So can I see your laboratories by myself as an observance student?
Of course. I think we can arrange that. Maybe not nowadays because of the pandemic.
Yeah,
but
because pandemic.
I mean, if you get I can give you my contacts later on. If you can reach me, we can at least have a coffee, and then I will try to show you around, and we can arrange something.
I'd love to. Thank you.
Yes, you're welcome.
Okay, thank you, Nilbeste. We're going to share, I guess, the contact of Gonca with for those who wants to get in touch with her. So there is a question in the chat part. It is written in Turkish. I'm not going to translate it.
I can answer it, shall I?
Okay, sure.
So shall I answer it in Turkish, in English? I don't know. But the question is, I was once upon a time in the HLA. The question comes from Miray Bonbon. And she said she's been in HLA lab in Cerrahpaşa Medical Faculty. And there they look at the compatibility between the patient and the donor using a microscope. So that's correct. That's also one method that we can match. It's called complement-dependent cytotoxicity assay. So we match, we combine, let's say, the patient serum with the donor cells. But now there is a technology that you don't need the donor cells. There are, let's say, microparticles. And if the donor cells attack these, if the patient serum attacks these donor cells, by adding a special dye and a complement factor, we can see which cells are lysed under the microscope and score it. So this is a very, let's say, conventional, but still very valuable method. It's called CDC. Very nice. So there is also one more question, if I may go on. So again, from Miray Bombom, is this, you're welcome, Miray. Is this screening system transplant immunology? So this is a very valid question, because when I look at my title, of course, we wanted to make it catchy for all you guys, but it's also a little bit not very clear. Maybe transplantation immunology is the topic that deals with the, within this transplantation field. So it is the part where we look at if the immune system is matching between the patient and donor. So it's called transplantation immunology. Within this transplantation immunology, I try to focus on how can we improve the matching. So transplant immunology is the field that I'm working on.
Okay.
Any other questions from my high school friends, other high school friends here? I'm wondering. They are also very, very, curious people. Zeynep Güngör, Çiçek Samioğlu, do you guys want to ask questions? You always ask me when I'm not working. Okay, well, they don't want to.
Okay, maybe we can have some questions from Serap Hoca. Serap Hocam, are you there?
Ben çok teşekkür ediyorum. Aslında Nilbes de benim adıma da soruları sordu zaten. Güzel sunumunuz için de çok teşekkür ediyorum. İngilizcemin yeterli olamamasından dolayı da özürlerimi diliyorum. Gerçekten çok faydalı ve güzel bir sunumdu. Öğrencilerimiz de biraz geç katılmak zorunda kaldılar. Dersleri vardı. Ama çok faydalı oldu. Çok teşekkür ediyoruz.
Çok teşekkür ediyoruz Serap Hocam. Biraz Türkçe'ye dönelim. Pardon. Biraz Türkçe'ye dönelim isterseniz. Gonca'ya sözü veriyorum.
Evet. Serap Hocam teşekkürler. Ben öncelikle benden İngilizce sunum istendiği için İngilizce konuşuyorum. Yoksa Türkçe konuşmaktan da zevk alıyorum. Dediğim gibi inşallah anlatabilmişimdir derdimi İngilizce'de de. Teşekkür ediyorum.
Çok güzeldi. Öğrenciler için de çok faydalı olduğuna eminim. Zaten gerek lisans öğrencilerimiz stajları sürecinde de bu tarz laboratuvarlara gidiyorlar ve bu tarz deneyleri deneyimliyorlar. Dolayısıyla onlar için de çok faydalı oluyor. Ben bu sunumların lisansüstü öğrencilerimiz için de çok faydalı olduğuna inanıyorum. Çok güzel bir süreç başlattı Zeynep Hocam. Devamının da güzel geleceğine inanıyorum.
Evet, çok teşekkür ederim. Sağ olun. Ben Türkçe'de yaparım eğer istenirse.
Evet, soru cevap bölümünde Türkçe devam ediyoruz. Evet, ben böyle ismen hocalarımı çağırmaya devam edeceğim. Serap Hocamı çağırdığım gibi. Evet, Kutsal Hocam nasılsınız?
Sağ olun Zeynep Hocam. Gonca Hanım tekrar elinize sağlık. Gayet güzel bir sunum oldu. Ben çok teşekkür ediyorum hem size hem de organizasyon adına Zeynep Hanım'a ve Murat Hoca'ya. Böyle güzel bir sunum gerçekleştirildiği için. Gerçekten öğrencilerimiz için bulunmaz bir nimet bu tür imkanlar. Gonca Hanım sizi tekrar yürekten tebrik ediyorum. Çalışmalarınızda da başarılar diliyorum. Sağ olun.
Teşekkürler Kutsal Hocam. Kutsal Hoca da merkez laboratuvarımızın yöneticisi, müdürü. Eminim hani böyle bir kolaborasyonlar
Tabii. İnşallah ileride
Gelecek
yapacağız.
kişilerden. O yüzden böyle bir yönlendiriyorum.
Tabii tabii. Çok
Ayrıca
teşekkürler.
burada Uluslararası Ofis Koordinatörümüz Murat Hoca da var.
Çok teşekkür ederim Gonca Hanım. Muhteşem bir sunumdu. Biz tabii öğrencilerimizi K107'de staj bağlamında yurt dışına da gönderiyoruz. Çeşitli üniversitelerde, hastanelerde de staj yapıyorlar. Belki sizinle irtibata geçerim sevgili Zeynep Hocam sayesinde. En az 1-2 öğrencimiz staja Erasmus bütçemizle, kendi hibelerimizle sizlerden destek alırlarsa belki Serap Hocam'ın öğrencileri olabilirler. Biz çok mutluluk duyarız. Sizlerin de böylece desteğini almış oluruz. Eğer uygun görürseniz tabii ki.
Ben tabii elimden geldiğince benimle iletişim kurulursa bir planlama dahilinde yardımcı olmak için elimden geleni yaparım. O konuda elimden geleni yapacağımı buradan söz veriyorum. Ve benim
Çok
dışında
teşekkür
gelişen olayları bilemeyiz ama bu pandemi geçtikten sonra
Tabii.
mutlaka bir yol bulunacaktır.
Evet. Çok çok teşekkür ediyorum.
Evet,
Zeynep
teşekkür
Hocam, Murat
ederim.
Hocam sizlere de öyle muhteşem yani. Çok zevk alarak izliyoruz.
Teşekkür ederim.
Ve gerçekten çok net. Ben hani matematikçi olarak çok dışında olmama rağmen Murat Hocam'ın dediği gibi anladım bir şeyler. Bu beni çok mutlu etti özellikle.
Ben
çok
de bir
teşekkür
şeyler
ederim.
anladığımı hissediyorum Murat Hocam.
Evet.
Ama yani şurada resmen 4 sayfa not aldım. Olur da Gonca bize soru sorar diye.
Eyvah eyvah.
Bayağı iyi öğrendim.
Alın var olun.
Biraz belki hani şu Türkçe kısımda hazır bir sürü lisansüstü master doktoru öğrencisi de aramızdayken bu kariyer petini oluşturma da nasıl yol aldığını belki Gonca'ya yöneltebiliriz. Tavsiyelerini almayı isteriz. Hani ikinci doktoru neden yaptığıyla ilgili bir şeylerden söz etti. Ama bu işte hani postdoc süreci veyahut da doktoraya kabul alma süreci, o bağlantıları nasıl kurduğu, yani bütün master doktora öğrencilerin aslında zihninde bu işi nasıl başaracağı, kariyerini nasıl hani o noktaya getirebileceği. O anlamda da paylaşımların çok güzel olur Gonca. Dinliyoruz.
Evet, konuşma esnasında da bahsettiğim gibi ben 11 yıl kadar İstanbul Tıp Fakültesi'nde çalıştım. Yani araştırma görevlisi olarak başlıyoruz orada, başlıyorduk işe ama oranın öncelikli kısmı hani bu organ nakli immünolojisi hizmetini verelim. Ardından buradan edindiğimiz deneyimlerle araştırmalarımızı yapalım şeklindeydi. Dolayısıyla bana kattığı deneyimi ben herhangi bir Avrupa'daki laboratuvarda öğrenemem. Yani o kadar iç içeydik ki klinisyenlerle, o kadar iş yüküne rağmen o kadar çok şey öğrendim ki bir laboratuvar çalışanı olarak diyelim. Orada dediğim gibi doktora tezimiz bizim mesai saatimizi bitirdikten sonra ilgileneceğimiz kısım oluyordu. Yani ister istemez insan rutin işin tatmini de tabii çok yüksek. Yani insan kendini çok iyi hissediyor böyle bir şeyin bir parçası olduktan sonra. Fakat dediğim gibi bazen insan içerisinde böyle anlayamadığı bir araştırma merakı oluyor. Ya bilmediğinden araştırma yapmanın ne kadar zor olduğunu, daha doğrusu prospektif yani ileriye dönük yeni fikirleri laboratuvar ortamında test etmek gibi bir heyecanım vardı benim. Standart rutin yöntemleri sürekli artık gözümüz kapalı uygulamanın dışında. Dediğim gibi bir tanesinin tatmini çok yüksek. Yani akşam eve gittiğinizde bir işin ucundan tutmuş oluyorsunuz. Birilerinin organ nakli olmasına vesile oluyorsunuz. Öte yandan da böyle garip bir araştırmacı olma isteği. Benim tabii tıbbi biyoloji anabilim dalındaki hocalarım, orada çalıştığım arkadaşlarım, bize öne yok olan işte Mahmut Çarın Hoca, Fatma Savran, Oğuz Hoca, bunlar hep yurt dışı bağlantıları olan, yurt dışındaki kongrelere öğrencilerini götüren hocalardı. Yani bence bu benim büyük bir şansım. Ben bu konferanslardan bir tanesinde şu anda çalıştığım merkezde şu an emekli oldu ama kurucularından olan Franz Klaas denen hocayla tanıştım ve yani İngilizce tabii bilmek çok önemli bu anlamda. Bilimsel olarak ilerleyebilmek, bilimsel olarak iletişim kurabilmek adına. Bence her şeyin temelinde zaten iletişim yatıyor yani. Ve ben bu yurt dışında tanıştığım, bizim de İstanbul'daki bu bölüme çok faydası olan bu hocaya, kongrelerde tanıştığım bu Hollanda'daki hocaya araştırma yapmak istiyorum dedim sadece ve Yani bunu işte her sene 10 sene boyunca kongreler oluyorsa, buradaki Türk hoca bizi götürüyordu oralara. Her seferinde söyledim, ben araştırma yapmak istiyorum, ben araştırma yapmak istiyorum. En sonunda gel o zaman dedi yani. Tabii ben böyle çok doğru adımlar atabilen bir insan değilim. Belki çok düşünüp taşınmıyorum ama biraz daha böyle içimden gelen sese kulak veriyordum o zaman, gençliğinde verdiği belki heyecanla. Bana dedi, sana bir doktora öğrencisi pozisyonu ayarlayayım, gel o zaman. Ben ne kadar para alırım, ne yaşayabilir miyim Hollanda'da, hiçbir şey düşünmemiştim. Bu fırsat bana verildi, aman Allah'ım ne kadar mutluyum diyerek, benim Çapa'da da zaten İstanbul Tıp Fakültesi'nde de doktoramdan sonra artık orada kalıcılığım kesindi, yani devam ediyordum. Ama bu istek o kadar baskın çıktı ki hemen 3 ay içerisinde bu kararımı İstanbul Tıp Fakültesi'ndeki hocalara da söyledim. Onlar da yani tabii ki senin için en iyi olanı biz de istiyoruz dediler ve bu şekilde yurt dışına gittim. Yani burada neden tekrar doktora yaptığımın sebebi, işin açıkçası bizim burada İstanbul Tıp Fakültesi'nde yaptığımız doktora farklı bir doktora. Yani biz hastalardan örnek almak için kendimiz çırpınıyoruz. İşte o işi halledelim, bu işi halledelim. Önce işimizi halledelim, sonra araştırmamızı yapalım. Halbuki araştırma aslında o bütün işi gücü bırakıp sadece odaklanılması gereken şey. Yani benim Hollanda'da elime geçen fırsat aslında bütün zamanımı sadece işte yeni bir teknik geçirmeye, geliştirmeye, yeni bir metot bulmaya adama fırsatını elde ettim ben. Tabii ki yani altyapı olarak inanılmaz bir araştırma bütçesi ve altyapısı var. Dolayısıyla yani bizim ülkemizde de tabii ki bunlar mümkün. Bunlar tamamen kişisel tercihler. Yani ben %80'ini rutinde, %20'sini araştırmada değil, hayatımın, iş hayatımın %80'ini araştırma, %20'sini rutin yapmak istiyordum ve açıkçası %100 araştırmada çalıştıktan sonra doktoram esnasında Hollanda'da. Tabii yani bir sürü tekniği, bir sürü bilgiyi öğrenmemiş gittim ben oraya. Hani doktora derecesini almak Türkiye'de başka bir çaba, Hollanda'da başka bir çaba. Yani biz burada 3 tane hastadan örnek alalım diye çırpınıyoruz. Hollanda'da biobank var. Yani hazır. Hiçbir şey yapmıyorsunuz. 100 hastanın örneği hazır. Orada daha çok düşünüyorsun işte. O şekilde yani tabii ki farklar var. Yani araştırma geliştirmeye önem veren, gelişmekte olan veya gelişmiş ülke farkı o anlamda ortaya çıkıyor. Dolayısıyla benim ikinci doktoramı yapmamın sebebi tamamen kendi eğitimimi Avrupa seviyesindeki araştırmacı eğitimine çekmekti. O konuda eksiğim olduğunu düşünüyordum. Deneyimimin fazla olmasına rağmen. Açıkçası bu ikisini harmanlamak bana çok faydalı oldu. Yani uzun lafın kısası, hocalar öğrencilerini yurt dışı kongrelere ya da uluslararası kongrelere Türkiye'deki göndersinler. Kendileri gitmesinler, öğrencilerini göndersinler. Öyle hocalar vardır çünkü sırf kendileri giderler. Bizim hocalar, Türkiye'deki hocalar öyle değildi yani, benim Çapa'daki hocalarım. Beraber götürüyorlardı bizi de ve tanıştırıyorlardı. Bu işte bu, bu işte bu. İngilizce de bildikten sonra yani ben yurt dışına gidip de başarısız olmuş bir insan görmüyorum ama yurt dışında yaşamak zor. Sadece başarı değil hayatta yani. Mesela güzel kokmak erkekler için de önemli. Bugün bunu öğrendik. Ya da işte doğru kokuya sahip olmak. Yani o tarz şeyler de var sonuçta. Dolayısıyla iletişim kurun. Kimsenin de yani aman pozisyonu şuymuş buymuş diye çekinmeyin. Siz iletişiminizi kurun, isteğinizi belli edin ve de kararınızı da verin. Yani kimse de ne olur buyurun Gonca Hanım gelin falan demedi. Ben de kalktım. Yani normalde rahat bir hayatım varken hayda tekrar başla öğrencilikten ve doktora yapanlarınız ya da yapmamış olanları çok moralini bozmuyor mu? Doktora da yani bir çile aslında yapmak, hani ikincisini yapmak da benden işte bu hale geldim sonuçta.
Gonca Hanım, şimdiki öğrenciler biraz daha şanslı. Yani dediklerinize %100 katılıyorum. Ben de Paris Saclay Üniversitesi ile çalışıyorum. 15 yıldır yaklaşık gidip geliyorum. 4 tane öğrenci mezun ettim oradan. Evet. Çalışmalarımızdan dolayı. Yani ama öğrencilerimiz biraz daha şanslı bizim dönemimize göre. Elle tutulur bir hocaları var yanlarında. Yani ben elinden tutup uçak biletini de alıp ödüyorum. Paris'e götürüyorum. İmkan sağlıyorum. Yani biraz şanslılar. Size hak veriyorum. Evet. O şeyimizi işte bulunmaz nimet dedim. Yani öğrencilerimiz için gerçekten altın bir fırsat. Öğrencilerimizin mutlaka bu şeyi de değerlendirmesinde fayda var.
Mutlaka hocam, harika bir şey yani ben tabii onu çok görmüyordum kendi zamanımda, böyle elinden tutup da götür. Benim hocalarım beni götürdü, o yüzden ben tanıştım. Ama şimdi mesela bu Erasmus'lar bilmem ne bu tarz değişim programları da var. O zamanlar o kadar yoktu. Zeynep benim yaşımı söylemedi ama ufak ufak ortaya çıkıyor tabii.
Estağfurullah.
Yani o zamanlar böyle fırsatlar yoktu. Bir de bu iş hep networking yani nerede olursanız olun, ne işte olursanız olun. Yani iyi İngilizce bileceksiniz maalesef yurt dışında. Ben çalışkanlıktan hiç şüphe etmiyorum. Yani oraya gidip de bu işi yapmayacak, kıvıramayacak arkadaşım yoktur herhalde. Ben yaptıktan sonra herkes yapar. Ama dediğim gibi iletişiminiz iyi olacak, kararlı olacaksınız. Kimseye de soru sormaktan çekinmeyeceksiniz. Mesela benim yani herhalde en zorlandığım kısım kendimi soru sormaya alıştırmak oldu toplantılarda. Bizde soru sormak misafire biraz acaba yani yanlış mı anlıyordu en azından benim tarafımda. Hocaya saldırıyorlar mı falan. Halbuki bakıyorum şimdi yani artık öğrendim. Soru sormak aksine ilgi duyduğunun göstergesiymiş. Aksine merak ettiğinin göstergesiymiş. Herhalde bence o aynı zamanda tetiklenmesi gereken en güzel şey öğrenci topluluklarında. Yani ders vermek değil de tartışmaya açık olmak, soru sormaya açık olmak. Sonuçta kimsenin de hiçbir şeyi başkasından daha iyi bildiği falan yokmuş yani.
Kesinlikle,
Onu da
kesinlikle doğru.
anlamış oluyor insan yaşla tabii.
Evet, çok aslında anlamlı şeyler de söylüyorsun. Belki biraz bizim bu seminerleri düzenlemeye başlamamızın altında yatan motivasyonu da bizle paylaşmış oluyorsun. Biz de burada istiyoruz ki yani hani şu lisansüstü öğrencilerimiz farklı disiplinlerden gelsin. Burada senin gibi başarılı araştırmacılar, akademisyenler veyahut da işte sektörden gelen kişiler konuşsunlar. Böyle bir tartışma ortamı oluşturalım istiyoruz. Yani bilimsel bir tartışma ortamı oluşturalım ve hani böyle bir herkese bir yol gösterelim istiyoruz. Çok teşekkür ediyoruz Gonca. Yani paylaşımların için harika bir konuşma olduğu gibi bence sonunda söylediğin şeyler de çok anlamlıydı. Herkes için de çok faydalı idi. Çok teşekkürler.
Ben teşekkür ederim.
Başka soru var mı acaba? Merak ediyoruz. Yoksa eğer seni böyle herkesin adına ben şöyle bir alkışlıyorum. Çok teşekkürler tekrar. Bütün katılımcılara da çok teşekkür ediyoruz. Sevgili hocalarımıza, sevgili high school friends, onlara da çok teşekkür ediyoruz.
Ben de buradan anneme babama da teşekkür edeyim. Onlar da katılmayı başarmışlar bizim Zoom toplantısına. Sizlere de çok teşekkür ediyorum. Ha? Açtım, duyuluyor herhalde. Ben de buradan hepinize çok teşekkür ediyorum. Çok keyif aldım. İletişimde kalalım. Ben elimden geleni yaparım. Türkçe'de konuşuruz gerekirse. Annemle babama da buradan merhaba diyorum. Bağlanmayı başardılar. Herkese öpüyorum. Hoşça kalın.
Evet, herkese sevgiler, selamlar. Görüşmek üzere. İyi akşamlar diyelim o zaman. Hoşça kalın.